First off, I would very much like to say thank you all for joining us for this talk that Ian summarized very well called Life After ChIP-seq. We're really going to be diving into the role of conformation and how that really enhances our view of the regulatory landscape, really in reference to ChIP-seq data that's generated throughout many different aspects of your research. So to give you a brief outline of what it is we're going to talk about today, I will be going over a brief introduction of chromatin conformation, what that means, how we generate those data, just so everybody's kind of on the same page. Then we're going to have a case study where we investigate the regulatory interactome of iPSC cells that are developing into neuronal stem cells. So really getting into the loss of pluripotency and how neuronal networks start to develop as these cell types differentiate. And finally, we will close off with how you would go about integrating this interactome data type in your research with a protocol called HiChip and what the data analysis kind of looks like there, just so you can kind of get a sense of how this data type would plug into the research that you're already doing today. So let's get started. We'll kick it off with that introduction into chromatin conformation. So one of the things that we like to start with in orientating people to is the fact that many epigenetic assays capture features in linear space, right? These are heavily rooted in primary order features such as base methylation, histone modification, or DNA protein binding sites, as well as chromatin accessibility. Now, one of the things that all of these data types have in common is they are sequence-based, but they all result in this linear image of the genome where your genomic position is on the x-axis and the enrichment or depletion of particular signals, whether that be methylation, histone modifications, or transposase accessibility, are enriched along that linear landscape of the genome. And that information is really helpful and has really bolstered our understanding of what I like to think of as the mechanical engineering of our genomes, right? The blueprints are all in the base modifications, but really how these genes get turned on and activated is very much directed at the epigenetic level. But one of these things that these assays don't tend to take into consideration is the fact that genomes aren't organized in a straight line. Now, if you think about the human genome, that's about 3 meters of DNA that has to be compacted into each nucleus per cell. So that's a lot of folding that has to occur to get everything nice and compact and structured inside that genome. And these features range a wide degree of sizes. So if we start at the very largest feature type, we're looking at chromosomal territories and how are these chromosomes actually compacted within the nucleus. And then if we go in and look at a particular chromosome, we can actually see that there are areas of the chromatin that are open, active, are associated with active gene transcription. Those are also called A compartments. And then there are B compartments, which are made up of heterochromatin, silenced chromatin, this is often referred to. These areas are often transcriptionally silent, so not active. And then if we go in and look at these compartments, we start seeing another level of organization. Now, these are called topologically associated domains, or TADs. And really what these TADs do is they kind of cluster linked function genes in physical space. So things are getting, a series of genes are getting activated and acted on and transcribed in sort of a similar fashion for that linked function. And then you can zoom in even further and start asking questions about promoter-specific interactions with transcription factors and other regulatory elements like enhancers that really are the key indicators to recruit, pull to, and kick off gene transcription. And so really what conformation does, this 3D folding of the genome, moves us from this very linear view of the genomic landscape when it comes to transcription, where you're thinking about post-translational modifications like acetylation and methylation, remodeling complexes, and transcription factors and enhancers being laid out on this linear format into what's a more realistic view of the genome in the 3D orientation of it, where we can actually start seeing now what enhancers and regulatory elements are interacting with their target promoters. Many times we don't know, if we're just looking at this linear format, which enhancer is actually interacting and acting on specific promoters. With this three-dimensional view of the genome, we can now really move from, again, this linear view into this three-dimensional view, where we can now associate those regulatory elements with the promoter of the gene in question here. So this is really powerful information, and we'll show you exactly why that is in our case study as we move forward. So how do we generate this image? How do we go from linear to three-dimensional? Well, it's through the world of chromatin conformation capture. This technology has been around for about 20 years, and there's a variety of different flavors that are associated with it. But the one thing that they all have in common is that they're rooted in this core workflow, where chromatin is cross-linked inside the cell in situ, and then that chromatin is digested. Historically, that's been done with restriction enzymes. We here at Dovetail have moved away from that primary sequence to a more chromatin-centric view, which is using MNase to digest that chromatin. And then from there, we perform proximity ligation, where these free ends ligate back, and instead of ligating back to what's closest in linear space, they ligate back to things that are close in three-dimensional space. And that creates this chimeric molecule, where the chimera event represents a physical distance that's being spanned by that junction. And then we reverse those crosslinks, leaving us with purified chimeric DNA that's reflective of those proximal associations between various loci. Now, once you have this chimeric molecule, you could do a whole variety of things to that chimeric molecule. You could do a qPCR approach. This was 3C. This was the original form of chromatin conformation capture, and that's where 3C technology really gets its name. Then you could do a 4C approach, where you take this chimera and you circularize it, and you know that you're interested in the blue region, but you don't know all of the interactions that could be occurring at this red site. So you perform inverse PCR on your known site, and then you amplify that region. And so now you're left with areas that are flanking your known region, so you can say what is associating with that blue region. What are these red unknowns? You could then, or you could perform a hybrid capture approach, where you have a handful of known loci. You design probes to capture all the various red regions, and you pull down on those, and then you ask the question, what are the different molecules on the blue side that are interacting with my sites of interest that are red here. What's commonly been most adopted in the chromatin conformation world is known as HiChip, in where this chimeric event has been biotinylated. Then you do a streptavidin enrichment on that biotin, and then you just convert that into a sequencing library. So at any given loci, you can ask what molecules or what other regions are interacting with any loci across the genome. Then there are more protein-directed approaches, and this is really kind of where we're going to focus today, where once you have your chromatin cross-linked, you can go in and pull down with an antibody on a protein of interest, allowing you to really target the rest of the proximity ligation events to a protein or a histone modification that you're particularly interested in. And there's a lot of benefits associated with that, which we'll get into at the latter part of our talk. So what do these data look like? Well, They're often represented on a contact matrix where each dot here would be representative of a read pair, where if we're looking at this dot here, read one of the paired in read maps here, read two of the paired in read maps here, and then the color of this dot would be the number of paired in reads that are associated with these two regions interacting with each other. So that's a cartoon of the data. This is what the data actually look like, wherein you can see that there are regions that are interacting closely where these two parts of the chromosome are interacting quite frequently with each other. And then even more so if we get closer in distance, that these two regions are interacting very highly. Now, there are regions that just don't interact that much, say these two regions, and that's how we start creating this three-dimensional image of this is a topologically associated domain, and this is a boundary between this domain and this domain. And so that's kind of how we build up that image of those conformational features. Now, if we bring in a more protein-directed view, that kind of changes how we view the data a little bit. So many of you might be quite familiar with these contact matrices by reading the literature. But when we bring in what's called a HiChip or a plaque-seq, which is really combining a chromatin immunoprecipitation with that proximity ligation information from HiC, what you get is a protein-directed HiC. So it's really the same core workflow. Instead of moving from fragmentation into ligation, you perform that chromatin immunoprecipitation and then conduct the ligation cross-link reversal and then convert that into a sequence library. Now, what that gives us is this view of the protein DNA binding sites with their histone modifications and how frequently those proteins interact with each other, or the protein DNA binding site are interacting across the genome. So here's normal ChIP-seq for CTCF, and you can see all the peaks there of where CTCF is binding to the DNA. Now, with this HiChip approach, we're able to recapitulate all of those peak signals, but now you get this added dimension of being able to see what two peaks are interacting with each other very frequently. For example, these two peaks interact with each other very weakly, whereas these two peaks interact with each other very, very strongly, given as seen here by the very strong green links connecting these two. And likewise, this peak and this peak are interacting very strongly here. And as we get into the case study, we'll kind of talk about how you would interpret that in terms of biology. So that's just a brief introduction into the world of HiC, how those data are generated, and how that data really enables us to move from this one-dimensional view of the genome to bring in this interactome data type, this three-dimensional view, to really give us the interaction frequency across the genome. So now let's get into our case study, the fun part, the biology, right? So to really set the stage here, I'm going to kick it off with the Waddington landscape, right? This has been around for many years, and it's a way to describe how you can go from one genotype to many phenotypes. So many people are familiar with the idea of pluripotency, where there's one genotype represented in a stem cell, and this stem cell has the potential to be differentiated across this Waddington landscape with ridges, with troughs and valleys that are often captured in the epigenetic signatures. So as cells start going down the differentiation pathway, there are different epigenetic features or sort of actors there that shift where a cell is determined to end up, whether it's going to be a neuronal cell or a liver cell or a muscle cell, things like that. And so this sort of landscape is really the landscape in which our case study works on. And so what we've done is we really wanted to ask the question, what role does that chromatin conformation or that interactome data type play in development and cellular differentiation? And it's been shown in the literature that at least up to 36% of differentiation in terms of chromatin is being driven by these topology changes, these conformation features. And so what we did was we looked at these iPSC, these pluripotent cells, and then we forced them to differentiate into neuronal progenitor cells, these NSCs. And we asked from a transcription, a ChIP-seq, and a HiChip, that interactome data, how are all of these, or how does that interactome data really capture this differentiation between these iPSC, this pluripotency, into a neuronal cell type? So to get started, we simply looked at the transcription, the RNA-seq data between these two cell types. And so what we're plotting here is the Go analysis between NSCs and iPSCs. And all of the dots here represent the number of genes associated in each Go function. And the color indicates how enriched that was in terms of a p-value. So the redder the dot, the more significantly enriched that is. The bluer the color, the more it's actually enriched in iPSC over NSC. And so what I'm calling out here is simply that in this upregulated NSC group here, that many of these features are either associated with development or specifically tied to neuronal function. So if you kind of just go through here at this green dot, this is nervous system development. If you look here, this is cellular differentiation. And across the board, almost all of the upregulated Go categories in NSC are associated with cellular differentiation, or particularly with neuronal development, which is great. That simply just means that the experiment that we've set up is working as it should. There's nothing groundbreaking here, but it's good confirmation to know that our iPSC cells look like iPSC cells and our NSC cells are behaving like NSC cells. So next, we ask the question from a normal ChIP-seq scenario: what is the enhancer, promoter, and topological boundary marks associated with these two cell types? How much is overlap, and how enriched are these signals? So for promoters, we looked at the H3K4 trimethyl. For enhancers, we looked at H3K27 acetylation. And for topological boundaries, we looked at CTCF. And so what you can see here across the iPSCs is that for the promoters and enhancers, there is a clear shift in where active promoters are being positioned at NSCs compared to iPSCs. Well, there is a significant amount of overlap in the promoter category. In the enhancer category, these two have very much shifted apart, and where a lot of enhancers that are present in NSCs are not necessarily present in iPSCs. But what is interesting here is that of the CTCF boundaries, these topological boundaries, there are many conserved regions, and there's not a great deal of difference between the boundary locations in NSCs, the neuronal cells, from the pluripotent cells. So we're going to talk about how we can kind of make heads or tails of this data in terms of that HiChip, that interactome data type. And so what I want to do next is point out the limitation by just looking at the ChIP-seq data type. So what ChIP-seq gives us, and if we're thinking about enhancers in this case, what it really provides us here is where the H3K27 anchor is. It tells us its position on the linear genome, right? On that reference genomes. It just tells us that, yes, there is enrichment of the lysine acetylation, or the 27th lysine acetylation at this site. But what it doesn't tell you is at what part is this acetylation acting on. And what the interactome data does, that HiChip data now will allow us to say, okay, here is where that enhancer signal is, but now we can link it to a genomic feature or promoter that's some distance away, right? And we can look at that and say, okay, now I can say that this enhancer is influencing this promoter. And this becomes really powerful if you're thinking about super enhancers that have many different promoters that they're interacting with. So you can actually categorize the sheer amount that each enhancer mark is actually impacting on various or a single promoter. So just to give you a sort of dummy experiment to see how you would interpret these data, we have this mock experiment set up where we have our gene track with two promoters, and we have an experiment, A and B. And in experiment A, this promoter is activated as represented by the RNA-seq data enrichment that you see. But in sample B, you see that both promoters have transcripts associated with them. And so your next question is okay, is there an enhancer mark that is gained or lost between these two? And in this example, we're saying no, the enhancer mark stays the same between A and B. So let's go to the Interactome. Let's see what HiChip tells us about these data. And so in the sample A, we can see that there is a link between this enhancer mark and this promoter. And we do not see this link between this enhancer and this promoter that's downstream here. Whereas with sample B, we can see that, yes, there is indeed two promoters that this enhancer is interacting with, which explains the transcription of this promoter in case B, right? So that's just a dummy view of how you would read these data types. So now let's get into the real data where we compare those iPSC cells into NSC cells. So first, we're going to look at the pluripotency maintenance factor NANOG here. And we're going to look at this from the enhancer, promoter, and topological boundary factors of H3K27AC, H3K4-trimethyl, and CTCF respectively. And so the first thing to point out here is that, is that there's a loss of enhancer and promoter contacts in NSC. So here we're looking at iPSC at the top, NSC at the bottom, and here's the enhancer mark, here's NANOG right here. And what we can see is while there is still an enhancer here and here, we lose this enhancer signal here, but we also lose these contacts that are present in iPSC that go away in NSC. Moreover, if we look from the promoter view going towards the enhancer, we see that yes, there is in fact an active promoter mark here, and we can see that it is in fact interacting mostly with this enhancer signal, not so much these signals here. But that signal is totally lost in NSC. Now, the other interesting thing that we can see in these two samples is a loss of these large topological features. So here are our CTCF that typically associate chromatin boundaries in folding. And what we can see in the iPSC is that there are these very long range interactions between this CTCF and this cluster of CTCF downstream here forming this boundary. That is lost in the NSC. So this boundary is now going away. Moreover, we could zoom in even further and say that even this sub-TAD signal, this sub-boundary signal, is completely lost in the NSC. So not only do we see a change in that enhancer and promoter marker, we also see that there is a breakdown of chromatin structure of that chromatin folding between iPSC that's lost in NSC. And what's even more interesting is that loss of boundary flanks the enhancer promoter interactions that are associated with the upregulation of NANOG in iPSC over NSC. So if we keep on this same sort of pluripotency interrogation here, we're looking at OCT4, which again is really there to help maintain pluripotency. And again, we see a similar pattern where we see a loss of enhancer promoter contacts. Again, here's your here's your enhancer signal, here's your promoter signal, and you see that the enhancer signal shifts between the two cells, but you also lose contacts here, and then here's POU 5F1 is also known as OCT4 here, and we see that over this gene, the interactome signal also changes greatly, and again, we can see this, this loss of interactome at the enhancer and promoter level also reflected in the loss of chromatin boundaries that again are associated with this OCT4 region that's again upregulated in iPSC over NSC. And so just to make sure that these data make sense, we went in and we performed normal ChIP-seq on NANOG and OCT4. And what you can see between iPSC and NSC is that the NANOG and OCT4 signal are very much diminished in the neuronal stem cell, but very much enriched in the iPSC. Just simply, stating the fact that these pluripotency maintenance factors are very much present in the iPSC cells that are maintaining their pluripotency and that are lost during the differentiation into neuronal stem cells. And so what I hope this kind of sheds some light on is just that looking at that one-dimensional ChIP-seq view might not be telling you the full story of the interactome and the regulatory factors that are interacting with the promoters that are regulating genes. Now, to take this one step further, we decided to take those neuronal stem cells and think about the enhancer interactome in light of Alzheimer's disease. So what we did was we looked at the ChIP-seq, the regular ChIP-seq data, and we overlaid that with GWAS identified Alzheimer's SNPs on chromosome 19. And as you go across chromosome 19, there is a huge enrichment of polymorphisms that are associated through GWAS with Alzheimer's. And so we wanted to ask the question then, does the regulatory interactome have some implications here in how we interpret SNPs in these data types? So we zoomed in on this high degree of polymorphism region, and we zoom in and we ask, okay, where are the enhancer signals across this region associated with a high degree of SNPs? So we did that, and then we asked, where are those promoters located? And then we really sort of dialed in and said for ApoE, which has really been shown to be implicated in the early onset of Alzheimer's, we really wanted to ask the question from ApoE's point of view onto what are the enhancers that are interacting with ApoE. So then we looked at the HiChip data for H3K27 acetylation, that enhancer mark, and what we can see here is where the promoter is for ApoE, we can see that there are at least 5 distinct enhancer marks that all have interaction, a high degree of interaction with this promoter. So that brings to light a few different things here. And so if you're looking at SNPs that only align with ApoE, they could be impacting that gene body itself, or it could be impacting the interaction with these enhancer regions that are kilobases away from that actual SNP location. Alternatively, if you look at the SNPs that are associated with these enhancer signals, right, you can maybe then start to think about the idea that the SNPs that, again, are occurring kilobases away from ApoE might actually disrupt the enhancer signal or the ability for acetylation to occur at those sites and alter those binding motifs, which now diminish the acetylation and the enhancer effectiveness of these regions that are interacting with the ApoE promoter. So from either direction here, it's clearly more complex than just linking SNPs that are associated at that gene. It's a very complex interactome where several different enhancers could be interacting and impacting the regulation of ApoE. So what this really indicates is there's a lot more research that really needs to be done to really pinpoint the mechanism of action that these SNPs have and within their relation to ApoE. And this goes across really any and all GWAS data, right? We know that there are many SNPs that occur in non-coding regions. There was a really great paper by Knott et al. In Science a couple of years ago that used a very similar data type to reassign GWAS SNPs to their actual gene that they're impacting through this Interactome data type. So it's when we talk about life after ChIP-seq, that's really kind of what we mean is you know where those signals occur on the genome, but you don't know really what they're interacting with solely based on that ChIP signal alone. And going for the nearest neighbor, for example, if we just take these two promoters that are enhancers that are close to the ApoE promoter, it's actually really hard to say which one of these two would actually be regulating it, or if it's actually a enhancer that is further away from that promoter region. So again, it really just kind of indicates that there's a lot more research to really dive into to really make sense of all of the GWAS data and all of the ChIP data that's been generated for these cell types. So now that you have kind of seen that Interactome data in action, let's think about how you would integrate that. And like I mentioned earlier, the kind of crux to integrating it is this HiChip assay by combining the HiC and ChIP-seq workflow. So I'm not going to go over this too much, kind of already went through it. It's the same core HiC workflow where we add in a chromatin immunoprecipitation. So you're really targeting your interactome with regions that you're interested in. But we do something a little different than the rest of the world at Dovetail in that we are nucleosome centric. We perform our chromatin fragmentation with MNase. And what this does is it gives us a very, very targeted view of the protein DNA interaction that you're interested in. So, like I was mentioning earlier, historically, chromatin has been fragmented with restriction enzymes. And this actually brings up a really big challenge in interpreting these data. So, for example, if we're going to look at, say, this hypothetical H3K27 acetylation mark on this particular histone, what you're actually going to get if you use restriction enzymes is you're going to be enriching your data here, right, at the restriction site, not actually over the histone in question. To further complicate matters, if you were to pull down on, say, this H3K4 trimethyl methylated histone here, you would be enriching at this restriction site and this restriction site. It would actually be rather unclear as to which nucleosome has been methylated. So our approach of fragmenting to the nucleosome level to perform that chromatin immunoprecipitation actually gives us a very nice tight view of the interactions and the potential proximity ligation events or that interactome data for your histone in question or protein binding site in question. Another added advantage that we see of using the MNase is that it almost is a 3 data type in one approach. So like I mentioned, you get your ChIP-seq signal, you get your interactome signal, but if you zoom in, you can now position nucleosomes around your feature of interest. It's almost like adding an MNase-seq assay to your ChIP-seq assay and then combining that with the proximity ligation. So these data are very rich and you can ask many questions with this particular data type. And I'll get into how you do that here in a few slides. One of the things that we do like to point out here is that there is a way that HiC approach where you're looking at the entire genome and not enriching over proteins, you can ask those same questions, but it's going to cost you a lot in sequence dollars to get there. And so here what we're looking at is a metagene analysis of those contact matrix. So think of it as we're taking all the CTCF sites across the genome, and we're just averaging the contacts across all those sites. And what you can see with the HiChip data at 150 million read pairs is this very nice punctate signal suggesting that CTCF sites interact with each other with a high degree of frequency, as denoted by that really bright spot. Whereas the off signal, like that off CTCF target signal, is very dense. So you get a very high signal to noise ratio in your data with a low sequence amount. Well, if you were to build those contact matrices to call chromatin loops, CTCF mediated chromatin loops with HiC data, you would have to sequence well over 800 million read pairs if you're using restriction enzymes like 1.2, 1.6 billion read pairs. And what you would get is, yes, you can kind of see that there is a increase in frequency of interaction between these CTCF sites on average, but it's associated with a lot of background noise. And so really you get more clarity with lower sequencing if you really target in your proximity ligation data to a protein DNA binding site of interest. So to integrate this, what feels like a complex data type into your work isn't actually as hard as it feels. It can be a little daunting when you're moving from that linear landscape to that sort of three-dimensional or connectivity approach. It's really not so bad. There are many tools out there that help process these data at Dovetail. You should definitely check out our GitHub page. We have very simple bash scripts that you can go in, open up, and take a look at to see exactly how you generate these data types. But we simply start with a FASTQ. We align using BWA mem to your reference genome to get your BAM file. Then we use a tool called Pair Tools, which is a 4D nucleome data type key here that really just boils down your BAM file into read 1 maps to this region, read 2 maps to this region, and this is the distance in between those two. And then you build your contact matrix, which basically says between any two region, count how many reads I see that occur that link these two loci. And that's the fundamentals of going from a FASTQ into a contact matrix. Now, from that contact matrix or your valid pairs file or your BAM file, you can do a lot of different analyses. So you can perform linear analyses. So just looking at coverage for that like MNase-seq profile type, or to get your ChIP signal, right? You can just use deep tools, BAM coverage, plot that in IGV, or plot that. I'm a big fan of the bioconductor package Sushi. You can plot it there. You can do pileups just like you would do with ChIP-seq. If you remember that NANOG signal, how we showed the average over all the NANOG sites between NSC and iPSC, right? Deep tools compute matrix or SandTools mpile. Those work great in building up those sort of views of the data type in one-dimensional space. And then you can perform peak calling with max2. HiChipper also has max2 built into it if you want to use that approach. And then when you get into more topological based analyses, like calling significant interactions in between your two peaks, HiChipper works well, HiChip works. There's a number of tools out there. Please feel free to reach out for us. We have a whole table that we can share with you on how to integrate different file types into different tools that are out there. The contact matrices work well with Juicer APA. The Cool Tools works really well with the contact matrices. There's a really nice cool Python tool out there called Cool Puppy that will allow you to build those average contact matrices with very little effort on your part to get that up and running. And then for plotting, I tend to just take the significant loops that are called from either HiChip or HiChipper and the coverage in a bed graph that you get from the BAM coverage in deep tools. And then I can just plot that in Bioconductor fairly easily. Many of these tools have outputs to use the WashU genome browser. So these are highly integrative data types. And so really the point here is just to not really be exhaustive in all the different ways you can analyze these data, but just show you that there's many tools out there that can help you process these data. And it's really not too much of a challenge to go from your BAM into that three-dimensional space. So before we close out here, I just want to point out that this is our probably most up-to-date version of our antibody list that we've validated. The part numbers and the suppliers of where you can get those. This list is always growing. We're always interested to hear in what you all are working on in the field. And if you don't see your antibody of interest, you should definitely reach out to us. We can give you some guidance on how to get started with your particular antibody of interest. So with that, I would like to say thank you very much to you all for listening, and thanks to everybody at Dovetail who contributed to this work.